So a bit of that. It's a bit like the Human Genome Project, which was sequencing the human genome or DNA sequence. The Human Cell Atlas aims to essentially profile all of the cells of the human body, and in the adult human there are about 37 trillion cells. And the idea is that you have then a reference of what are the cell types that are present in the human body. It was launched very recently, in fact, late 2016, and this was at the Wellcome Building. And essentially this is an aggregation of many researchers from many institutions across now 83 countries who are pooling their efforts to identify what cell types comprise human tissues, organs, and the human body. Anyone can join, so you just have to click on that link. The fundamental approach is essentially, this is all at single cell resolution. And the earliest technology that was scalable was being able to measure the gene expression profile of an individual cell. And that has now escalated, rapidly evolved, so that you don't just measure the gene expression, you can measure the gene expression as well as some surface protein and also open chromatin. And all of that gives you a profile of what a cell is, and you can use that profile to then identify its cell type or give it a label, and then really then begin to understand how all of these cells make up the human tissue. I'm going to focus my talk on the developmental cell atlas, and really this is focused on studying human development, which is extremely difficult, has really been a black box up to now. Much of what we understand has been shaped by looking at animal model systems. But to illustrate, and you've heard lots of the talks all throughout today and this afternoon where I've been here, the many disciplines that have to come together to bring in the expertise to not just drive the technology, but also the understanding of what these biological systems are telling you, and how these measurements are now not just multimodal, but also across scale. You're moving from single cell and being able to then put that together into how the tissue is comprised and how the organs and the human body eventually. And this is going to be quite difficult to achieve because most of science has, particularly biological sciences, has focused on where you have individual groups working on individual projects. And what you really need to do is where many people, many groups come and to work one project. The effort is very different, the approach is very different. You need to have the infrastructure to be able to do this, the research framework to be able to work collaboratively in team science fashion, using the most cutting edge technologies, and how are you going to make the best use of the data and also disseminate the data. And that's something that has been made possible by having this global initiative within the Human Cell Atlas. So to just frame a bit of the biology behind what I'm going to talk about, and that's how the immune system develops. The key point from this slide is that it occurs across many tissues and across gestation time. So you have this space and time that you have to somehow stitch together when what you're doing is really making snapshot measurements at specific gestational time. And in the UK, we're also very lucky. You've heard about all these resources. There is another one, which is the Human Developmental Biology Resource, funded by the MRC and Wellcome, one based in Newcastle and one based in London, which really support researchers who are wanting to study human development. And it's quite important that we don't just base this on what's known in animal models. I've put here that in the mouse, pretty much everything that, the blood and immune system is contributed by the fetal liver hematopoiesis, whereas in human, you have bone marrow hematopoiesis emerging from about the second trimester of pregnancy and really dominates from later gestation and after birth. So what we did was to essentially profile all of these individual organs, all of the cells, to have an idea of what cells there are and the environment that they're in, and we put all of that information together for the individual organs. And finally, we integrated all of that information in a recent publication. I've got that as bioRxiv, but it came out a few weeks ago, which is also available for anyone to read. So the idea, by measuring all of the cells, is to then be able to kind of construct the framework of the immune system in this case, but you can imagine it as any organ or any organism for that matter. and begin to see the whole as being greater than the sum of its parts, and beginning to appreciate the emergent properties that you wouldn't otherwise see if you were to just analyze just a few cells or distinct lineages. And this is very much a work that has been brought together by individuals from many disciplines, not just my group, involving immunology, hematopoiesis, developmental biology, and computation. And as I mentioned earlier, this is the final manuscript that tries to integrate all of that information, which we published recently. The data is all available to download as either data objects or the raw data itself from repository. And you can just click and it tells you exactly what type of organs there are. And also all of the code, for example, is available so that you can actually reproduce the analysis that has been done. And it's usually, there's a Zenodo link at the bottom for reproducibility. We also provide the data in the form of a web portal. Two of the other speakers have also talked about web portals. Here you can look at the expression of genes in the context. This is CD20 on the B lineage, and you can interrogate the data and look at many other parameters, including the types of clinical metadata and so on. And more recently, we included the expression of genes that are known to cause diseases in human. And in this context, because the data is fetal bone marrow, we looked at genes that are implicated in inborn errors of immunity and childhood leukemia. So you can then just click on the data object. It will take you drop-down menu of search for diseases, and you can search for all of the different diseases that are there, and it gives you the genes that are implicated, and you can look at the expression, and it will tell you which cell type is actually expressing that particular gene. A bit of what we discovered with regards to the developing immune system. The punchline is in human, this is completed by 20 post-conception weeks. In the early stage where fetal liver dominates, what you see is everything except neutrophils. Neutrophils are the most abundant white cell after birth. And what you see in the fetal bone marrow is the emergence of neutrophils alongside eosinophils and basophils. So some of the highlights, which I think is really important in terms of, these are observations, these are biological insights, and how do we understand what are determining these observations and insights that we've made? One is how the hematopoietic stem cell actually changes over gestation time, which you can probe not just by the molecular or gene expression profile of those cells, but also functional assays, where if you take these individual hematopoietic stem cells and ask what cells can they differentiate into, and then you can actually compare the cells across gestation and across two tissues that are forming this hematopoietic function. So in the first, you can look at hematopoietic stem cells across gestation from the fetal liver. And what I'm showing here is the fact that if you took hematopoietic stem cells from a very early liver, its ability to differentiate into the erythroid or the red cell lineage declines over time, but its ability to differentiate into the B lineage or myeloid lineage increases over time. So telling you that, just very quickly across several weeks of gestation, you see these differences. And then if you took the same hematopoietic stem cells from matched fetal liver and bone marrow, you'll find that their properties are also different, in that the fetal liver are much more supercharged and can proliferate better, and they're also much better at differentiating into multiple lineages. So you can learn a few other things about the distinct lineages when the granulocytes appeared, as I said, and when some of the dendritic cell subsets diversify within the fetal bone marrow. And if you then start to compare it across age, including adult bone marrow, you can see how this composition changes across the human lifespan. And also begin to appreciate some of the more unique features of the immune system. In particular, what you could call it is like moonlighting features of immune cells, in that we know them as cells that are important for immunity, but actually during early development, they play a critical role in tissue morphogenesis. And antigen presentation, which is what we know them for, really only emerges from the second trimester. One of the interesting highlights from this study is the presence of progenitors primarily in the fetal bone marrow, and this relative proportion is extremely contracted in the adult bone marrow. And this also is why infant and childhood leukemias are primarily B lineage because you've got the progenitor substrate. And also the discovery of new cell types, particularly B1 cells, which have only been described in the mouse, not in human. And we can then use this type of data sets, looking at the features that have been described for these cells in the mouse, and show that there are indeed the equivalent of these cells. They are like the innate lymphoid cells for T cells. So they are more proliferative. This is the proportion of cells that are cycling, and they spontaneously secrete antibodies, primarily IgM, both here shown in the form of RNA and also protein by ELISpot assay by isolating the cells. Another discovery from the T cell perspective is if you can then look at the thymus and look at how all of the different T cells are made in the thymus and ask the question, what are the T cells that are found in the fetal bone marrow? They correspond to the differentiated T cells that have left the thymus and then seed the bone marrow. So they're not made in the bone marrow. And another really incredible discovery from the perspective of unconventional T cells are that they're found in a wide variety of organs only during development, and that what we know as organoid models that are making T cells by culture of induced pluripotent stem cells are not conventional T cells, but in fact unconventional T cells. So all of this information, really helpful in terms of how can we better engineer tissues, cells for therapy, and also ultimately for tissue engineering. So those are the kind of incredible insights that you can get from just studying this by profiling individual cells to very high resolution. But you can also now use this healthy reference to map alterations in disease. And some diseases, particularly affecting infants and children, develop during susceptible windows in prenatal life. And one of them being Down syndrome, a chromosomal alteration, whereby you have, trisomy 21, so 3 copies of chromosome 21. And this chromosomal alteration results in altered hematopoiesis and also the potential of developing megakaryocytic myeloid leukemia in infant and also during early development. And you can then use these sort of unsupervised ways of analysis, not just by kind of identifying what cells there are by looking at their gene expression, but also how the abundance of those cell types and how the qualitative and quantitative changes occur in one condition, i.e. Down syndrome, compared to non-Down syndrome. And you can then begin to look at how having an extra copy of chromosome 21 affects the multiple lineages at single-cell resolution and also by every chromosome, which I've not gone to show here. Another way that you can use this type of reference data is to take, for example, in this case, we took infant and childhood leukemia bulk RNA sequencing data. You can then ask for these leukemic cells, what is the signature of those leukemic cells and how they map to the different cell states that we've identified in the fetal bone marrow. And for a small fraction of the infant leukemias, and these have an aggressive phenotype and prognosis, the signature that was primarily dominant in the bulk RNA sequencing data was not of differentiated B cells, but actually of early lymphoid progenitors. And then you can interrogate the RNA and protein data. In this case, we had also generated site-seq data, which includes both for individual cells. Find the combination of surface proteins that allows you to target these cells in a selective way and advance sort of new ways of treating this condition. Another example of how these types of analysis actually tells us a lot more about how diseases occur and how personalized medicine can be informed in the future, which I'll come to a bit later, is knowing that or beginning to understand that some of these developmental programs also re-emerge in inflammatory diseases such as skin diseases. And this was something where we compared healthy skin with developing skin and with diseased skin. And the highlight of this study was the fact that the skin vascular endothelium, one subset of it, here V3, and macrophage 2, was transcriptionally very similar to many vascular endothelial cells in the developing organs, and the same for the macrophage 2 and developing macrophages. But what was really amazing was that these two subsets were increased in lesional skin of patients with atopic dermatitis and psoriasis. And you saw that not just in our single-cell data, but also when you deconvoluted bulk RNA sequencing data from existing data sets. And beginning to understand what is going on, these vascular endothelial cells are like high endothelial venules in the skin, and they express a lot of adhesion molecules. And if you look at the gene expression program that's conserved between the lesional skin cells with developing skin cells, they're all about leukocyte recruitment and angiogenesis. So essentially, during development, you have this program that is switched on to allow leukocytes to enter the tissue, and it's also the same program that later becomes switched on to bring immune cells into the tissue to cause inflammation. So that's what we propose, and we highlight in the manuscript the different receptors and ligands that are involved in this process. But what we don't know is whether this pathway is regulated in a similar way during development and during disease, and if it has any other relevance in other inflammatory disorders outside of skin. I mentioned about how the way that the kind of infrastructure and the framework of operating for Human Cell Atlas really gave us a lot of expertise and the know-how to kind of work together collaboratively. And when COVID-19, the pandemic occurred, we very quickly mobilized to try and understand the immune response in COVID-19. And the only thing that I want to really illustrate is that this needs to sort of like be accompanied by the recognition that when people come together, you do need to have plural recognition of contributions. And we're beginning to sort of like just say, why not recognize as many people who are really involved? Because to be honest with you, you can't really deliver this type of work in 4 months, analyzing 130 patients, 1 million cells, and really kind of like getting from generating data to full-blown manuscript, and providing the data in available objects for people to download and also to browse. But the reference atlas was really critical in the early stages of the pandemic, because these were the first few papers that really told us where the receptors for COVID or SARS-CoV-2 entry receptors were, and not just in the nasal epithelium, but also highlighting the conjunctiva and the salivary glands as potential access points. So I'm going to finish now, but really to just say that this type of approach is really important in terms of highlighting insights and for tissue engineering and cell therapy, beginning to understand chromosomal alterations, childhood disorders, and how the understanding of human development is relevant for adult pathology, and really working very quickly in terms of coming together as a big group, particularly in COVID-19. And in terms of precision medicine, I think the field that I've described to you is really going to interface with precision medicine and so on, because as we scale up and we can start doing these type of analysis on lots of patients, and as the computational approaches really develop to begin to integrate multimodal patient data and across scale, this is going to be extremely powerful and not just going to be determined by genetics. It is going to be bringing together and giving completely different insights. And we saw a little bit earlier about how one gene protein can have many different ways of affecting diseases that haven't been put together in one basket in the past. I think this will begin to revolutionize. So this is something that our fields are going to emerge and work together, hopefully in the near future. If not, it's already started now, I guess. And just to emphasize the importance of kind of doing this collectively and bringing in different perspectives, but also the need to provide infrastructure and framework so people can work together. There's an incentive to work together, to have the recognition and acknowledgement of products and outputs that have been achieved as a collective. So I'm going to thank my lab for the fantastic years of research and to acknowledge my many collaborators and our funders. And thank you very much for your attention.